Welcome back to the Deep Dive. Today we're digging into a really interesting machine learning paper. It's tackling cardiomyopathy diagnosis using endomyocardial biopsies.
Yeah, this is a tricky area. These biopsies are invasive, right? And getting enough samples, well, that's tough. Plus, interpreting them needs real expertise, and there's a shortage of cardiac pathologists.
Exactly. So this paper is from Nissan University, and they're asking, can we use machine learning, specifically looking at the texture of these biopsy images to help diagnose different types of cardiomyopathies more reliably?
It's a neat angle because, a lot of the buzz in medical imaging is around deep learning, CNN specifically.
Right.
But CNNs, they need tons of data. And for these biopsies, well, you just don't have that kind of volume.
That's the core problem they're addressing. With small data sets, complex models like CNNs can overfit pretty badly. They learn the training data too well, noise and all, and then fail on new cases.
So this paper takes a step back almost. They're saying, okay, let's use, let's say, more traditional ML models, less complex ones, but let's be really smart about the features we feed them, specifically these texture features.
And they want to know two main things. First, do these texture features actually work? Can they separate, say, hypertrophic cardiomyopathy from dilated cardiomyopathy or from normal tissue?
Mm-hmm.
And second, given that we have this small data set problem, what's the best model design? Like, how should we combine feature selection, maybe dimensionality reduction, and the actual classifier algorithm?
They looked at quite a spectrum of conditions too. Hypertrophic, dilated, restrictive, even rarer stuff like cardiac amyloidosis, sarcoidosis, Fabry disease, compared against normal cardiac tissue.
So maybe we should talk a bit about the history here. Why texture features now?
Yeah, good idea. I mean, for ages, diagnosis was purely down to the pathologist looking through a microscope. Highly skilled, but subjective, and likely had fewer experts available.
So computational pathology started trying to help out.
Right. Early attempts were maybe a bit basic, simple image processing. The real challenge was always how to quantify what the pathologist sees, how to get numbers out of those complex tissue images that actually mean something clinically.
Before things like texture analysis got sophisticated, people were kind of stuck handcrafting features, weren't they? Trying to measure specific things they thought were important.
Exactly. It required a lot of domain knowledge, and you might miss subtle things. That's where these more automated texture analysis methods come in. They look at statistical properties, pixel relationships, repeating patterns, stuff the human eye might not consciously register or at least quantify consistently.
They use 6 different methods in the paper, right? First order stats, GLCM, GLRLM. Bunch of acronyms.
Yeah, the whole toolkit, basically. FOS looks at the simple histogram properties, mean brightness, variance, skewness, that sort of thing.
Okay.
Then you have methods like GLCM, the gray level co-occurrence matrix, which looks at pairs of pixels. How often does a bright pixel appear next to a dark one, for instance? That gives you contrast, correlation.
And GLRLM.
Gray level run length matrix. That looks for runs of pixels with the same intensity. Are there lots of short runs or long runs? It gives you features like short run emphasis, long run emphasis.
ADF, RDF, angular, and radial distribution functions.
Those are looking more at like directional patterns and frequency content within the image texture. All told, they calculated, 39 features?
Yeah, 39 distinct texture features. A lot to work with.
Definitely. So the first step after extracting them is usually normalization or standardization. They used a robust method here.
Why robust?
Well, medical images can have artifacts, outliers. Robust standardization helps make the features less sensitive to those extreme values, puts everything on a similar scale without being skewed too much by weird pixels. It's pretty standard practice, but important.
Okay, so you've got 39 standardized features. Now what? You can't just throw all 39 at a simple model with only, say, 60 samples.
All right. Exactly. That's where feature selection, FS, comes in. They used a method based on variance, trying to find features that have low variance within a class. So all normal samples look similar for that feature, but high variance between classes. So normal looks different from diseased for that feature.
Makes sense. Maximize the separation.
Right. They calculated this variance ratio for each feature individually, which is computationally cheaper than looking at combinations. And for the multi-class problem, they broke it down, compared each pair of classes, found the best features for that pair, and then took the union of all those best features.
Clever. So after selecting maybe a smaller set of promising features, they then looked at dimensionality compression, or DC.
Yeah, using Fisher's linear discriminant analysis, LDA. LDA is supervised, so it uses the class labels to find a lower dimensional projection, like squashing the data down from, say, 15 dimensions to just 2, in a way that keeps the classes as separated as possible.
Why only 2 dimensions in their case?
LDA's output dimensionality is limited by the number of classes -1. They had 3 main groups: disease, borderline, normal. So C1 gives you 2 dimensions. It's a common choice for visualization, too.
Got it. So features extracted, maybe selected, maybe compressed.
Yeah.
Then classification.
Then classification. They kept it relatively simple here. Support vector machines, SVMs, with both a linear kernel and an RBF kernel.
Which are kind of workhorses in ML.
They are. And decision trees, DTs, which create sort of axis-parallel decision boundaries. Lower complexity models, generally better suited for smaller data sets compared to, say, a massive neural network.
And how did they make sure the results were reliable? Testing on the same data you train on is a big no-no.
Absolutely. They used nested stratified k-fold cross-validation. Bit of a mouthful, but it's a robust way to do it. You split the data into k-folds, train on k1, test on the held-out fold, and repeat k times. The nested part means they did this twice an outer loop for evaluating the final model performance and an inner loop for tuning the hyperparameters.
Hyperparameters, like the settings for the texture methods or the SVM.
Exactly. Things like the bin width for histograms, the distance used in GLCM, the SVM cost parameter, the decision tree depth. Lots of knobs to tune. They used Bayesian optimization within that inner cross-validation loop to search for the best settings efficiently. It's a pretty rigorous setup to avoid overfitting and get a fair estimate of performance.
Okay, so let's talk about their actual experiment. They had data from 6 patients.
6 autopsy patients, yeah. 4 with hypertrophic cardiomyopathy, 2 showing that classic cardiomyocyte disarray, so disease, and 2 without it, which they called borderline, and 2 patients without heart disease as the normal group. 10 samples per case, so 60 samples total. Not a huge data set.
No, definitely small. And they tested different combinations, right? Feature selection or all features, dimensionality compression or none, and the 3 classifiers.
Yep. They basically tried 12 different model pipelines, FS alone, DC alone, FS then DC, or just using all features, AF, combined with each of the 3 classifiers, SVM-LK, SVM-RBF, and DT.
What did they find? Did the texture features actually work?
Overwhelmingly, yes. They did statistical tests on the features themselves after finding the best hyperparameters, and 32 out of the 39 features showed significant differences between the groups. Things like the average pixel intensity, the contrast, variance. They clearly separated the classes.
So the texture is capturing something real about the disease state.
It seems so, although interestingly, some of the directional features from ADF didn't show strong significance. They thought maybe the orientation of the tissue slices wasn't uniform, which could wash out those directional signals.
And how did the different model designs compare in terms of predictive performance? What F1 scores did they get?
The performance was generally pretty good across the board, which is encouraging. But they saw some patterns. Using just dimensionality compression, LDA, on its own sometimes led to overfitting great scores on the training data, but a drop on the test set.
That can happen if LDA tries too hard to separate the training data when you have way more features than samples initially.
Exactly. SVMs tended to do a bit better than decision trees overall. But the star performer, in terms of generalizing to the unseen test data, was the combination. Feature selection plus dimensionality compression, FS plus DC.
So prune the features first, then compress.
That seemed to give the best balance, leading to the highest test set F1 scores, especially with the SVM classifiers. They got up to around 0.949 with SVM linear and FS plus DC.
Wow, that's impressive for such a small data set. Did using all features work badly?
Surprisingly, no. Using all features, AF also did quite well, suggesting many of the 39 features were genuinely useful. But the FS plus DC approach was just slightly more robust on the test set, which is what you care about for real-world use. It seems like that 2-stage reduction is really beneficial when your number of features swamps your number of samples.
So key takeaways. Texture features definitely have diagnostic value here. And this multi-stage dimensionality reduction, FS then DC, seems like a solid strategy for small, high-dimensional medical data like this.
Absolutely. It helps build models that generalize better, even if they're relatively simple models like SVMs or decision trees. And importantly, these models are potentially more interpretable than a big black box CNN, which is a plus in medicine.
Right. Doctors want to know why the model is making a prediction.
Precisely. Though they did note some instability in exactly which features got selected in the FS step, which isn't surprising with small data. But overall, a strong argument for this approach.
Okay, this feels like a good point to transition to our novel notions segment. Where could this research lead next?
All right, so building on this idea that texture is important, but acknowledging patient variability is key. Here's idea one: personalized texture feature signatures.
Intriguing. So moving beyond just disease versus normal texture.
Yeah, imagine instead of one set of best features for everyone, we learn a signature specific to each patient. You'd start with their initial biopsy, characterize its texture, but then maybe track how their specific texture changes over time, perhaps with follow-up biopsies or advanced imaging.
So using longitudinal data.
Exactly. You could use techniques like meta-learning or few-shot learning, which are designed to adapt models with limited data. The goal is to tune into the subtle, patient-specific textural shifts that signal their unique disease progression or how they're responding to treatment.
That tackles the individual patient differences limitation they mentioned. What's the big challenge?
Well, current methods often lump patients together. This needs AI that can learn effectively from sparse individual time series data, and integrating different data types like imaging and biopsy texture over time is non-trivial.
Probability.
I'd say medium. Personalized AI is a hot area, but the longitudinal data aspect, especially integrating invasive and non-invasive data, needs work.
Okay, interesting. Idea two.
How about generative modeling for synthetic biopsy data augmentation, but with texture control?
Tackling the small sample size problem head on with synthetic data.
Right, but not just any synthetic data. Using things like GANs to generate new biopsy images, but critically, conditioning the generation process on the key texture feature statistics we know are important.
So telling the Jan, make me an image that looks like hypertrophic cardiomyopathy and has this specific mean intensity, this level of contrast, these run-length properties.
Exactly. You're leveraging the knowledge from this paper about which texture features matter. If you can generate realistic images that match the texture profiles of different conditions, you could massively expand your training data set, especially for rare diseases where real data is almost impossible to get in large quantities.
That could be huge for model training. What's the catch?
The obvious missing piece is ensuring the quality and biological relevance of that synthetic data. It's easy to generate pretty pictures. It's hard to generate pictures that accurately reflect the underlying pathology in a way that genuinely helps the diagnostic model learn. Controlling those specific texture features is the key challenge.
Likelihood for this one?
I'd say high. Generative modeling in medical images is booming. Adding feature-level control is a very active research direction. People are already working on similar concepts.
Okay, cool ideas. And just the usual reminder, these novel notions are purely speculative, just food for thought.
Definitely. So wrapping up the paper itself, strong validation for texture features in cardiomyopathy diagnosis from biopsies, and a really practical demonstration that a smart dimensionality reduction strategy feature selection followed by dimensionality compression can make simpler ML models perform exceptionally well, even with very limited data. That FS plus DC plus SVM linear combo hitting nearly 95% accuracy is quite something.
Yeah, really significant work. If you want to dive deeper into their methods, the statistics, all the details, the link to the paper is in the description or show notes.
And maybe a final thought for everyone listening. As these AI tools get better at analyzing medical images like biopsies, how do we best integrate them? Not just for diagnosis, but could they help us spot entirely new visual biomarkers or understand disease progression in ways we haven't before? It's more than just automation. It could be about deeper biological insight too.